Good evening, everyone. How's it going? I shall introduce myself, being a member of the BVI committee. Thank you very much for coming along to the BVI seminar this week. My name is Martin Howe. I'm at the School of Biological Sciences working on sensory ecology, particularly visual ecology of animals. Before I start, I'm going to start a shameless plug to all please go and watch Our Oceans on Netflix. It's the latest blue chip production it's involved lots of people from Bristol. So I watched the first couple of episodes and it's absolutely fantastic. Narrated by Barack Obama, who's not a scratch on David Attenborough, but he does an all right job. Okay, so thank you for coming. I'm going to talk to you about cuttlefish because this keeps coming up at various meetings that we always talk about cuttlefish. But actually, I'm going to be a bit controversial and I'm going to start not talking about cuttlefish, but talking about crabs, because this is another love of mine. And I'm hoping that this will all come together at the end and you'll understand why I'm starting with crabs. When I first started off in science, I did a PhD in Australia, looking at these remarkable animals. These are fiddler crabs. They live out on the mudflat of Australia. It's a very two-dimensional environment and their sensory systems are really kind of adapted to living in a flat world. You'll see these males here have got big claws that they wave at each other, and the females, they tend to not have the big claws. They have smaller claws that they use for feeding. So these are the females here that you can see in the foreground. This is a little sequence that was shot by the BBC. They wanted to tell a love story here. So excuse the whole kind of cheesy leading up to this, but you've got a female here and you've got a male who's eyeing up the female and trying to court her. But the thing that I was studying was the motion signals that are involved in this courtship. So you'll see after a while, once they stop sort of batting their eyes at each other, the male is going to start waving its large claw. This is a communication signal that they use to try and attract attention and also to show who's the best. And also as a species identification cue, which I hope I'll show you in a minute. So they're about to start courting. Here the male starts raising the claw and lifting it up, does this incredible sort of semaphore display, which it waves towards the female, and the female uses that as a cue to help decide which male to mate with. I did my PhD in Australia at the Australian National University with some of these figures that you may recognise. There's Jochen Zeil, Jan Hemme, Johannes Zanker, who's from Royal Holloway here in the UK, and Jochen Smolka, who's now in Sweden. And what we wanted to do was to get into the mind of these crabs. We wanted to see what the females are watching when they're watching these males. So we built this camera system, very simple. This is old school digital 8 camera filming down onto a front surfaced mirror here at 45 degrees. And that let us film right down at the level of the mudflat with these females. And we could get beautiful video of these crabs waving. And then we could also downsample these videos into crab resolution. Here you're going to see a tern flying overhead in a minute, and you'll be able to see that this tern just appears as a flickering ommatidium in the visual system of the crabs. So we can really kind of start modeling what these crabs are seeing. But I was particularly interested in motion at that stage. What is it about the movement of these claws that is relevant to the females? And to do that, this is where we teamed up with Johannes Zanker. We had all these digital videos of waving animals, and we wanted to kind of process those through the visual system of the female and understand what the movement component was. So we use these models of elementary motion detectors, really simple systems looking for motion that between adjacent pixels in an image. And we simulated that across a whole digital image and then processed frames of video through that, pixel by pixel, and you can extract motion from a scene. So here's one of our crabs and it's raising its claw. And once we process that for motion, we get these motion maps. So this map is color-coded according to the direction of the border here. So the green area of the claw is moving in that upward direction. So frame by frame, you can pull out where the movement is occurring from the perspective of the female. You get lots of really nice motion maps. So here's an upward phase of a claw wave and a downward phase. And you can see it's got lots of greeny, reddy colors, whereas that's more bluey, downward purpley colors, and then a different species also. And we were able to then compare different species of claw waving. So here are a bunch of different species that we found on the Australian mudflats. and I've just got the XY position of the claw tip through a wave bout. And over time, this claw tip is either going up and down, or it's going up once and then down, and so on. But then we could also use the motion that we extracted from these digital videos to make something that looks a bit like a spectrogram that you would study kind of bird song with. But instead of using kind of the spectrum of sound, we're actually using the direction and strength of movement along this y-axis, and then time along the x-axis. So you can see this claw is flapping up and down. So you're getting strong up, strong down, strong up, strong down. You're getting this flapping sort of cue. This claw is kind of got a slow upward movement and then a downward movement. So we're kind of trying to quantify these signals a bit more from the perspective of the female. So we then sort of got interested in other sorts of motion in crab vision. We were interested in motion to do with gaze stabilization. This is a different motion vision task that most animals have, but crabs are great for studying. And this is essentially, if your body is moving, you want to stabilize your eyes relative to the outside world so you don't get too much motion blur, and it helps you to process the visual scene in various ways. So it minimizes motion blur. And you might have heard of the optomotor reflex. Certainly anyone who's involved in vision science will know about this. As you move your head, the eyes will snap to different features. And that's an optomotor, sort of the motor movement of eyes. And you can elicit this really nicely in the lab by making the world move around an animal, either with projectors or with an actual physical rotating drum. And that might look something like this. So here's the mouse in the middle of a cylinder. And if you rotate that cylinder, the pattern on the walls makes it look like the whole world is moving to this animal, and it'll try and stabilize its eyes. And this is what crabs do if you put them in one of these. So here you've got a top-down view of a crab glued to a stick. We do glue our animals to sticks quite often. You'll see in this reflection here, you can see that there's some stripes that are moving in the background, and the eyes are tracking those stripes and then snapping back, tracking those stripes. And right down here, we've got a plot of eye angle over time, showing that they're following that drum and then snapping back. And we can use that very simple behavioral paradigm to study all kinds of aspects of this sort of motion vision. For example, we had a visitor in our lab, Krista Up, who came and tested the optomotor reflex of these 4 different species of crustaceans. And if you test them with lots of different spatial frequencies of patterns, and lots of different contrasts, you find that they have this nice contrast sensitivity function. So there's a peak spatial frequency and contrast that their motion vision system works at. And these vary between species. So low resolution animals aren't very good at seeing these higher frequency patterns than ones that have much more sophisticated visual systems. We also managed to build a drum that we could make polarization patterns happen. So this is, instead of the intensity of light, we're actually playing with the angle of light waves. And these animals weren't able to do that task at all. It's like this motion vision task was mediated entirely by intensity with no input from their polarization vision. And that's analogous to what we know about other animals that can't use their color vision for this sort of motion-based task. Another important motion vision task, which is actually probably going to be the most relevant when we start moving on to cuttlefish later in this presentation, is predator detection. Okay, one of the main things, particularly if you're a crab, that you want to worry about is something that's approaching towards you, because that's probably going to eat you. Likewise, if you're a scuba diver, you do not want to see this thing expanding in your direction. So we've developed some protocols in our lab to play with predator detection with crabs. So this is Sam Smithers, who was a PhD in our lab for a while. And again, we took this fiddler crab that we can find down in southwest Spain, and we subjected it to, well, first one of the main predators that these crabs are susceptible to is birds. They're very worried about birds coming and eating them, because these crabs are out at low tide on the mudflat. Birds will hunt them from above and swoop down and grab them. So they're very interested in whether there's going to be an expanding stimulus coming towards them from above. And these panels on the left here show you just an example snapshot of what birds will look like in brightness. So you can see some birds, some parts of birds even look very high contrast, while others might have lower contrast, right? Especially when you start down sampling that to the resolution of a crab, which you have down at the bottom. So this is all they're seeing is really kind of very little information from far away. But this image here, this is a representation of the sort of contrast that's available in the polarization of light. So the background sky, which is color code, kind of got a false color code here, has a very high level of polarization, usually up to about 50 to 70% polarization. It's quite high. Whereas birds act as a silhouette against that. So there's very little polarization coming back off these birds. And even when you downsample right to very low crab resolution, you can still get some element of contrast there. And so we were quite interested in whether they use polarization for detecting predators. So Sam ran some nice experiments. This is one of our stock standard experiments that we run in the visual ecology lab. We have a treadmill and we'll mount a crab on top of the treadmill so that it's stationary in one place. But the treadmill can rotate underneath the crab. So the crab can walk or feel like it's walking, but it's actually staying stationary in one place. And then we can show that crab various things on screens, and we can modify those screens to show things in color or polarization or brightness. Here's what that sort of setup looks like. Here we've got an array of LCD screens, and then we've actually got a special screen here with a projector. There's a light source, so we can manipulate all sorts of elements of this. And this we can take into the field. So we set this up in our Airbnb room in the southwest of Spain, and we can just collect the animals from the mudflat, run them in the experiment, and then put them back on the mudflat after we finished. Here's an example scary stimulus that we use over here. This is our simulated predator. Very, very simple. It's just a disc that expands. And it expands in such a way that it stimulates that sort of reflex flight behavior of these animals. And here's a video of one of the crabs on the treadmill. So you can see this treadmill is supported on a cushion of air. So as the crab moves, the treadmill just rotates underneath it. And at some point there, you can see it freezes. So that's where it's seen one of the stimuli, and it's just frozen still. So it's a really clear behavioral response that we can score very accurately as to whether they're detecting a certain contrast of predator or not. So when we show them predators that are in intensity, we get something like this. So I'll just see if I can move this bar a bit out of the way so you can see the x-axis. So this is the Weber contrast. This is how much contrast there is between the predator and the background. So a contrast of 0 means that the predator is the same intensity as the background. If we go negative, then that predator is darker than the background. And if we go positive, then it's lighter than the background. You can see as you go further and further away from here, you're getting higher and higher contrast. The chances that the crab are going to react to that predator are increasing. So you see there's this nice sort of response profile as it gets higher and higher contrast. You also get that in the other direction, but it's not quite as strong. They're much more sensitive to sort of silhouettes than they are to bright objects coming towards them. And we see something very similar in polarization. So here we have instead a different estimate of contrast based on polarization, which we call polarization distance. And that's a model of what we think that the contrast is available to the photoreceptors in the eye of the animal. And you can see they react really well to polarized predators. So they're using their polarization vision also for this task. So both polarization and intensity information is contributing to this visual task, whereas it isn't for the gaze stabilization we saw earlier. And just to round off this section about crabs, there's some really beautiful work that's come out of a lab in Argentina, Daniel Tomsik's lab, which is looking at some of the neural sort of underpinnings of this behavior. And they found some fantastic data showing exactly which neurons are involved in detecting predators. So this is an image of the lobula of one of the optic neuropils in a crab. And they've measured from some of these neurons here and find that they are essentially loom detectors. So they have a visual field in part of the eye, and they are sensitive to an expanding cue, but not to other motion cues. And really coolly is that there's an array of 16 of these, and each one of those has a different direction of sensitivity, spanning the entire 360-degree range around the animal. And so if you map these, you've got these 16 different receptors that have a different position around in the lobula. But then also, if you map that as the azimuth position that they are facing, you can see that each one of these essentially is sensitive to predators approaching from different angles of azimuth around the animal. And these are wired almost directly up to the escape machinery of the animal. So if one of these loon detectors fires, then the animal runs in the other direction. And that running is almost directly coupled to the strength of the expansion of that field. So it's a really simple system that these crabs use to detect predators and to respond to them. And I want you to remember this when we come to talking about cuttlefish later. Another aspect of visual noise also that we've got interested in the lab recently is visual information, is visual noise. So these shallow water environments in particular can be really visually noisy. These are this is the caustic flicker that you get on the sea floor, which is caused by lensing of light as it goes through the sea surface. And so animals living in these sort of habitats have to cope with this quite challenging visual environment. And there's been some lovely work coming out of Bristol looking at the sorts of impacts that this sort of lighting environment can have on the behavior of animals. And so here, there's some data about sticklebacks. So they spend less time stationary in noisy environments, and they avoid spending time in noisy environments. So they much prefer to move away from those. And there's some lovely data also from Sam Matchett, also from Bristol at the time anyway, who was looking at the efficacy, the ease at which triggerfish can find prey items within different noisy environments. And so he had these lovely iPad experiments where he gave triggerfish, he trained them to find an object on here. You can just see it moving along here. And eventually it finds the object. And once it finds it, it gets a food reward. And so he tested different combinations of dynamic and static backgrounds. And these triggerfish could always find that target much quicker when the background was static than when it was moving. So dynamic noise in the environment does impact animals. It makes it harder for them to see stuff. So we had an excellent master's student a few years back, Sean, who was quite interested in this question. And we decided to ask this question of crabs. Can crabs, do they find it harder to find predators in a noisy environment? And so we used simulations of these sort of dappled lights generated by virtual reality software programs. You sort of generate sort of pseudo-realistic moving or static caustics, but very controllable. This is the sort of behavior that we see. This is the top view of the crab, and you'll see a stimulus appearing. And there you go, you get a nice reaction. So it's kind of, it can see that there's something there. This is a stimulus viewed against a static background at the moment. And then we also did the same for moving. So what do we find? Here's when you have no noise. So crabs, as the contrast of the predator increases, they find it easier and easier to react to it. Obviously, if there's zero contrast, then they have zero response. But when we add noise to the system, then they really don't do very well. So it really does negatively impact their ability to detect the predator. So yeah, background motion noise can be have a strong impact on these kind of things. But I then got quite interested in whether maybe there might be instances where predators or prey can exploit some of these aspects of visual systems for camouflage, either to improve their ability to catch prey or to improve their ability to avoid being preyed on. And I guess that opened up the can of worms that is motion camouflage. So is it possible for animals to somehow camouflage their movement? And there's a few examples of how animals might do that. One way might be to minimize the motion cues that they're generating when they're moving around. So something like this Australian strobe ant has a moving strategy where it dashes very fast and then it stays stationary, dashes very fast and stays stationary. So it's confining its movement to very short bouts. And then maybe when it's stationary, it's harder to find. So maybe there's movement strategies to minimize being detected. You can also try and blend into background motion. So here's an example of a vine snake, for example. It lives in wind-blown vegetation, and as it's moving, it has a swaying motion. So it's essentially trying to match the background of motion that it's living in. There's some quite cool examples maybe of dragonflies being able to do this when they're trying to intercept other competitors. They'll try and match the optic flow experienced by the target by flying in such a way that they look stationary against the background. So it's matching that optic flow that their target is experiencing. It's kind of cool. There's quite complex maths involved in trying to prove that that is really the case, but there seems to be some evidence for that. But then there's also this quite cool idea that maybe animals might manipulate motion by generating confusing motion cues, something to throw off a predator or a prey item. And we use the term dazzle very loosely, like it's not really very well defined. And I think actually this is an area of the field that could do with very, much better definitions to understand what is going on here. But immediately one of the examples that we jump to is zebras. So I'm going to segue from crabs to zebras and then to cuttlefish. So we were quite interested in maybe the zebras might be an example of systems that can generate these false motion cues. Like they're very conspicuously striped, really high contrast. They don't seem to be trying to camouflage to the background, although there's some evidence that maybe from a distance they might blend in and be harder to find. But again, I teamed back with Johannes Zanker, who I'd worked with during my PhD. And we spent some time looking at the sorts of motion cues that you might experience when you're looking at zebras, using some of these very simple motion detection algorithms. And we were quite interested in a few different optical illusions that might be going on. There's this wagon wheel effect or kind of aliasing where stripes get mismatched. And so you kind of detect the wrong sorts of motion cues. And then there's also this barber pole illusion where a moving edge looks like it's moving actually in a direction perpendicular to the stripe. And you can simulate these effects using these sort of motion detectors. They have limitations though, because we're dealing with video frames, which are very stroboscopic. They're kind of serial presentations, which is different from our own visual systems. But there's some things we could learn from that. I then teamed up with our illustrious Tim Carrow, because we wanted to get some evidence that perhaps these stripes might not be targeting predators, but maybe flies. There's quite a lot of behavioural evidence from decades ago even, that maybe zebra stripes might be conspicuous to avoid being landed on by flies. And so here we did some simulations of what a visual scene, this is a zebra being viewed by a horsefly that's coming into land, sort of downsampled to their sort of resolution. And then we generate a motion map, and then you can see that you get these very asymmetrical motion profiles. You don't get a nice expanding cue. We also went out to try and gather some evidence of what is the mechanism? Is it the stripes per se that are causing this problem? So we went out and luckily there's a horse farm just down in Dundry, just 10 minutes drive from here, where they have some zebras. So we watched flies behaving around zebras. We watched flies around horses dressed up as zebras. And we counted how many flies were landing on each. And sure enough, almost no flies landed on stripy zebras or horses. So there's a really strong effect of this zebra stripe pattern. And we tested a bunch of different patterns. And what we found is that actually any sort of high contrast pattern, whether it's kind of vertical stripes, horizontal stripes, or even a checkerboard, was actually quite effective at reducing the number of horseflies landing on them. So actually, zebras could have evolved to be checkered rather than stripy, it turns out. We also wanted to try and look at the precise mechanisms. What is it that's stopping these flies from landing on these horses? And so we used these stereo camera systems. These are 2 GoPros mounted on a bar, and we calibrate the system so that we can triangulate the position of flies as they're flying around these horses, trying to land on them depending on what pattern they have. And so we tracked the flights of a lot of these animals, and this is a sort of reconstruction of all of them simultaneously in one figure. They're over multiple days rather than all together. And we were quite interested in some of the flight dynamics. Is there something about how they're coming into land that might be telling us something about what they're doing? And we also tested a bunch of different patterns. So we had some checkerboard, we had a random check just to kind of mix things up a bit. We had something that was a bit camouflage, so it had an even number of black and white, but it was a bit more of a of a natural kind of pattern. We also tried making something that was dorsally dark and ventrally white, and vice versa, which also plays a bit with the polarization pattern that these horses are producing. And in summary, actually, what we found is that most of these high contrast patterns reduced the number of flies landing. It didn't seem to be anything specific about stripes, which was slightly annoying because we were kind of hoping that there'd be a really sexy sort of motion dazzle sort of mechanism going on here. And so we're still not entirely sure what the underpinning mechanism is, but horse flies are just as unlikely to land on this pattern, which doesn't generate so many of these sort of motion illusions as they are to land on, you know, something a bit more like this. So there's more of a correlation with sort of the contrast and the spatial distribution of that contrast in the animal. So yeah, we never quite managed to sew that one up, but we had some quite good insights maybe as to what it's not about anyway. So one example that I think I've come to now of a true sort of motion illusion, I think, is a species of cuttlefish that I first learned about in this documentary that I watched back in 2007. It's an Australian documentary about cuttlefish in general. And they had a short sequence in it, which was filmed by Dr. Mark Norman, but with a lot of interpretation from Roger Hamlin, two very important people in the kind of the history of cephalopod research. I'm just going to play the short clip. Once I saw this, I was hooked on this as a study system. I thought I've got to study what's going on here. But what we'll see here is a hunting cuttlefish. This cuttlefish is out looking for a meal. So it's looking around. This is in the reef of Indonesia in Raja Ampat. It's a very complicated reef environment. So it sees a crab, and then it does something really strange with its head. It pulses this dark pattern across its head as it's coming into this final moments of approach. It's these stripes, essentially, that's passing down its head, and it's all within the last sort of meter or 2 meters of approach. So it's definitely focused in on the animal, directed straight towards it. It's not produced on the back of the animal at all. This is something that is really directed towards the prey item. And so being someone who's quite interested in motion and motion camouflage, I'm like, wow, there's got to be something going on here. So along with Mark Norman, I teamed up with Mark Norman, and we went and surveyed a lot of the literature and found a lot of YouTube videos that amateur divers have made of this. And we got a few metrics out of it. This is a little metric that shows how fast the stripes move as they're coming into hunt, and then the prey escapes, and then the stripes slow down. So there's some dynamic aspect to how these stripes are produced. We did a few kind of stick diagrams of what these stripes look like, but we didn't get any further than that. Then we thought, actually, well, now we've already got to really try and find out what's going on. I'll introduce you to the species. This is the broadclub cuttlefish. It's the only species of cuttlefish we know that does this hunting pattern. And interestingly, it only seems to do it in the Indonesia area, certainly around Raja Ampat. You get the same species on the barrier reef, but people don't observe it doing this behavior. So it seems to be a regionally specific behavior. You can just see the variety of body patterns that they can adopt. They're really remarkable animals. They've got this adaptive camouflage and adaptive signaling that they can just change at the drop of a hat. They're really spectacular animals to work with. And they're the second largest cuttlefish. They're very big, which means you can't really study them in an aquarium at all. So they're actually really quite hard to study. And to bring it full circle to the crabs at the beginning of the presentation, they love eating crabs. So that's great. We've got a prey study system for which we know quite a bit about their motion vision. We've got a hunting animal that does this crazy motion-based pattern when it's coming into hunt. This is an ideal study system that we can really get our teeth into to try and work out what's going on. So first thing we did is we tried to understand what the prey is experiencing. So we went back to our good old crab on a stick treadmill, and with help from Nick Scott Samuel and students, this is Mubaraka who helped us with some of the very early experiments, and then Chris and Matteo who did the majority of the more recent work. What we did is we set up these crab treadmills, and we offered them stimuli based on what we were doing before, but with a motion component. So just again, this is what the crabs do. They sit on the treadmill, and when they see a predator, they freeze and they go still. And we offered them 3 different stimuli. We offered them something similar to what we've been showing before, just a simple expansion, an expansion with stripes on it that aren't moving, an expansion with stripes on it that are moving. And we gave them a whole range of contrast, ranging from no contrast to really, really contrasty. And we wanted to see whether those moving stripes did anything to reduce the response rate of these crabs. So here's what we found. So the first example, this is just a normal situation with a normal expansion. What we've got here is contrast along the x-axis. And so as contrast increases, the likelihood that the crab will respond to that stimulus increases too. So that's exactly what we'd expect. With the stationary stripe example, we get exactly the same. So this is an expanding cue just with stationary stripes on it. But then interestingly, when we gave them moving stripes, this actually had quite a profound effect at the high contrast end. So the higher contrast it was, the lower the probability the crab was of responding. So it almost dropped it by half. So even though this is a really, really obvious thing that the crab can definitely see, it's these big moving stripes, it's actually reducing the probability that the crab's going to respond to it. Super cool. Okay, it's one thing to stick crabs on treadmills. We actually really want to see what's going on in the field. So we were lucky enough that we got a few grants together, including a Marie Curie for Matteo Santon. And we went from the lab into the water. This required a lot of investment in scuba diving and rebreather diving equipment, as well as photography underwater and so on. It's exponential. The costs and difficulty of doing stuff underwater really increased exponentially. We also had Cedric Vandenberg in the lab at the time, who has spoken here before. So he's a very experienced diver as well and came and helped us collecting data. And again, we went to find the broadclub cuttlefish at this place, which was recommended to us by the BBC, who have filmed this species in this location doing the hunting behaviour that we were interested in. This is Cree Island in Raja Ampat, and this is where we stayed. It's an absolutely beautiful place to work. And occasionally we would find that all of the animals would actually be sitting under here, so we could just jump off the jetty and film the animals. It's really great. This is one of the animals that we worked with. You can see it's got they adopt the camouflage that's relevant to their background. This made it extremely difficult to find the animals in the first place. They're really, really well camouflaged. So we spent a lot of dives just swimming, trying to find the animals. Here's another example. This is a great example of kind of using the posture of the arms to match the posture of the coral that it's around. And then there's another one. You know, these are really quite hard to find. And really what we're looking for is that yellow blush around the eye and the slight undulating fin. And that's one of the ways that we could break their camouflage, but they're extremely difficult to find. And in the end, I think we got data from about 50 cuttlefish over 2 years, and that was 4 different research trips. And we managed to catch data from about 150 feeding events, which I'll show you in a minute. So what did we find? We did a few things. The first thing we wanted to do was to get the prey's perspective on this hunting display. So what we did is we mounted a GoPro right behind a prey item. We could only get this cuttlefish hunting to happen with a live crab. So we got special ethical permission to use live crabs, which we collected from the very abundant populations on the coastline. And this is the sort of thing that we find. So you've got a little crab who's sitting just down here, just underneath the lens of the camera. And you'll see here, the cuttlefish has spotted the crab and it starts approaching it. So it's doing a sort of a general rough sort of camouflagey sort of display, trying to look like it's not a cuttlefish. Then when it gets close, it's going to switch strategy. So it folds its arms forward into a cone and has these 2 other arms held out straight sideways. You can see it looks like a really long, thin line rather than a round approaching object. And then it's got this dark blush that it passes in a diffuse stripe pattern across the body. This is a female that's also got quite a lot of scarring on its head. You can see it's quite near the end of mating season. And then when you get close enough, there you go. So the feeding tentacle, really fast, ballistic stripe, goes out and it's got suckers on the end of the feeding tentacle, which then grab onto the back of the crab and lift it up. So the cuttlefish have to hit the crab at the right place. They have to hit it on the carapace so that the suckers can stick. And so they can pull it back into their mouth without getting nipped by the claws. The other bit of data we collected was using a stereo camera rig. So this is a series of GoPros on a bar, just like we were filming those horseflies with around the zebras and the horses. But this time we're doing it underwater. So we've got these simple kind of waterproof GoPros and we calibrate them so we know spatially where everything is. And we can feed the animal coming in, film the animal coming in and trying to feed on this, this crab. And you get footage a bit like this. We've got two slightly different perspectives on these feeding events. One slightly to the right, one slightly to the left. And that lets us triangulate the position of everything over time. And these GoPros can film at 240 frames a second, so we can get really good temporal resolution on this as well. We could reconstruct the tracks that these animals are coming in on and have a look at the dynamics of the speed that they're approaching the crab prey items. And we can also pull out some nice metrics from the video, like the size of the cuttlefish, the length of the feeding tentacles when they strike, the size of the crabs, and we'll look for correlations in all of the data as to whether there's a something to do with how they're using that stripe display. And then probably most interestingly is we used some of those motion detection algorithms to study the sorts of motion output that comes off these animals when they're coming in to hunt. And what we find, this is the distribution of motion vectors that these crabs, that these cuttlefish elicit from a crab's perspective when they're doing a stripe display versus when they're not. So you can see when they're not doing a stripe display, because they're quite long and thin, they elongate out these arms, you get kind of a good kind of up and down sort of motion cue. So it's not quite a proper expansion, but it's certainly a bit more symmetrical. When they're using these stripes, you get this huge downward motion vector. So the crab is really experiencing a downward motion field. And any of that expansion is being kind of overwhelmed, if you like, by that component. So that, I think, is telling. I think that's probably telling us that this is a true example of motion camouflage. So it's not trying to blend into the background motion. It's actually using a very conspicuous salient motion to overwhelm or camouflage that expanding cue, which is the thing that fires those escape neurons in the crab and makes them run away. So by making a non-threatening, overwhelming, high contrast movement, it's camouflaging the threatening approaching movement. So that suggests to us that this is a true example of motion camouflage. It's using motion to sort of somehow mask or overwhelm the motion cue that it can't avoid giving off. One of the other quite cool things that we came across while we were doing this is that these remarkable animals don't just use this as a hunting strategy. Now, this video sometimes glitches and stops, but I'm going to leave it because it does get going after a while. But what I want you to see here is we've got 4 of the same species here doing 4 different hunting strategies. There's the leaf, the stripe, the star, and the pulse. So this stripe display is the one that we've already been talking about. But they actually only do this about a third of the time. They also do these ones the rest of the time as well. So there's this very interesting motion display here, which we call the pulse, where the animal just hovers in front of the crab and it pulses this dark blotch of color up its up 2 or 3 of its arms that it holds out sideways. They do the more conventional sort of camouflage display, which we call the star. So this is trying to look like a bit of weed, I guess. And then this one we call leaf, because we think it's probably trying to masquerade as a floating mangrove leaf. And it actually produces this nice sort of counter shading on it to make it look like a flat leaf surface. So what we're quite interested in now is following up whether there are any things about the prey type or size or the conditions that they're doing these displays in that make them decide to do one display over another. At the moment, it's not obvious to us what they do. Sometimes you can give the same animal 4 crabs in a row and it'll do 4 different displays. So animals don't have a particular preference for one or the other. So yeah, we're hoping to get back into the field sometime and follow that up as well. So a lot of people contributed to this work, and there was a lot of funding from different funding agencies, and we're really hoping to get some more funding to get back out in the field and also to do some more lab experiments. But I'd really it'd be great to chat to everyone about motion camouflage and what might be going on. Thank you. I'll leave this one up because it's quite cool.
Really, really lovely work. I'm curious to know the pattern of movement that's going downwards. It's a little bit counter to what you think they might do. And it looks a little like an optomotor drum. And I wondered, is there any chance, have you filmed what the crab does? Because it seems like maybe the crab might kind of move downwards, exposing its carapace to make it easier to catch. Does that happen? Do you know?
We actually had that idea quite early from the beginning. We're thinking, well, maybe this is actually somehow driving the crabs to lean forward, which then exposes the carapace, which means that they can then get shot. I think we haven't been able to really look at certainly free prey and look at how they react to these things. We might be able to get something in the lab, maybe on a treadmill, but I don't know, maybe we could just try a really kind of big wide field optomotor experiment and see if we can actually get some behavior in the crab in a lab. It's very hard to do this with live prey in the field. In fact, the whole time we were there, we spent hours and hours with these animals and we only saw one natural feeding event. So either we're not getting there at the right times of day for feeding, or it's just quite rare. I mean, if you give them a crab, they'll go for it. So I think there's probably not much prey around. So I guess what we could try doing is releasing crabs near them and just try and film the crab behavior as the animals are coming in, rather than filming the animals coming into the crabs. But yeah, good call. That's it.
I have a question about the zebras. So, does the model assume that the horse or zebra or whatever animal is effectively completely still? So I was just wondering that if you're a fly, you're going around the zebra, your visual system can probably adjust to the fact that you're flying and things are happening fast. But the zebra is not completely still, right? It's, even though it's feeding, there might be a bit of micro motion going on. And I wonder whether that could have an effect into this whole system. That very slow motion.
It could do. I mean, we essentially modeled it as a stationary zebra or horse, because we're just assuming that these flies are moving fast, so most of the movement they're experiencing is going to be their own egocentric optic flow sort of base movement. When we were thinking about the predators, that's when we were thinking, okay, well, maybe there's microsaccades that the predator's eyes are doing that are flicking side by side, and that's causing aliasing or wagon wheeling or whatever. Or also, if you've got multiple zebras together and they're moving at slightly different rates, then you've actually got quite a visual challenge, because you're trying to track individuals within the field with stationary eyes, but with moving objects moving in different directions. So maybe it's particularly effective with herds of zebra, for example. I think we just wanted to collapse it down as simply as possible with the modeling and just model. It's literally just a straight line flight straight towards a zebra. We did have ideas that we could actually take the real trajectories of horseflies that we've been gathering and then model in a three-dimensional environment exactly what that horsefly is experiencing as it's coming into land and see if there's something going on. But because we didn't get a very strong indication that the stripes were really playing an integral sort of motion camouflagey role, we never went down that route. But it'd be quite fun to explore that in more depth for sure. Yeah.
Well, thanks for the talk, Martin. So for the top right one, a similar video to that earlier, you saw it kind of switch from this background matching approach with a similar posture after it's seen the crab, and then at certain distances switches to that. You know what kind of determines that distance when it becomes more profitable?
Yeah, we quantified that. So we were quite interested in when they decide to put on this display. And so we could, because we had these stereo cameras, we could track right from when they first spotted the prey. You can see really clearly because they're just hanging out and then their heads just go boom. So you know when they see it. So you know right from that moment, let's follow it coming in. And they only ever switch to that final hunting display within the last probably meter, sometimes slightly further, meter and a half. And before that, they adopt something else. It might be the standard, what this kind of thing. They might come in like that and then switch strategy just at the end. So it's really, I think, when you're a predator approaching, the most motion you're inducing is at the end. That's when your expansion is really gets logarithmic expansion. Sorry, exponential expansion very fast. That's when the biggest cues are there, and that's when they're using this display. They don't do it when they're further away. Nick.
So the hunting display looks a lot like a caustic pattern would look on a plain object moving in the water. When you've been jollying around in Indonesian seas, other predators of crabs utilising that, so preferentially attacking in caustic environments when there's wind on the surface? Does it look like they get the same benefit, right?
Yeah.
Projected on the plane.
So, yeah, I mean, these animals will do this anywhere between 0.5 meter depth, where you get really strong caustics,
Yeah.
and 20 meters where you got no caustics. They'll still do it. But you can see that there is direct sight, line of sight to the sea surface, and so there is movement behind the animal. So one of the things we were quite interested in is what the background looks like to the animal. Are they more likely to do this when they're seen against the top? And this when it's not motion or something like that. But you can see this one, there's really strong sort of dapple light there as well. We couldn't see anything obvious. And sometimes it's quite turbid as well. You just can't see anything. There's no caustics or anything. It's really diffuse light. So whether or not they're trying to sort of match that caustic background, there's not massive evidence for that. But whether or not other animals make use of that, I think there's a lot of quite interesting work we could do with that. You know, are there, especially if you've got times of the day where you've got caustics turning on and off because of variable wind conditions, maybe animals are actually conserving energy during stationary times and then coming out during
a fan, like you did with
Yeah.
You put a fan on the surface to induce caustics.
Yeah. And we thought of exactly doing that in rock pools, for example, down in Torquay. You could just have a rock pool and a leaf blower and kind of just track the animal's behavior just using long-term cameras and give them two different motion conditions. It'd be really interesting. Yeah.
It's funny because this animal has the ability to dramatically change its shape and size. And oddly enough, I mean, I used to try to sneak up on groundhogs when I was a kid. And the way to do it was to get smaller and smaller as you approached, because that's the opposite of what you would normally do. If you go sideways, they don't see the movement of your legs. It was very effective. I can get very, very close. These animals could be doing the same. They could start off big, and as they get closer, it gets them. But they don't do that, which suggests
They
that,
sort of do.
well,
Like,
the
watch
very,
this. You
in
see
the
it
very
again?
last second.
Can you see it's putting its arms
Yeah,
in just
yeah,
before? So they
just as it
sort
does
of
it.
do it, sort of just right at the end.
Yeah.
Yeah.
But I mean, some of the other ones definitely aren't doing that.
Yeah,
And it just feels like it was such an easy thing to do if in fact it was effective.
yeah.
But
yeah.
underwater, sometimes the way you judge depth might not just be size, it might even be contrast.
Yeah.
Because as you can see, as you get farther away, the contrast decreases dramatically.
Yeah.
So whether this is somehow playing into some kind of contrast
Yeah,
function
that's
as well.
true. I mean, the other thing that I think I'd like to explore is also shape. So these things are really long. When you get right behind the crab, they're just really long. And I'm wondering whether maybe if you're a predator that's approaching, maybe it's better to be long and thin than round, because the round thing generates kind of strong motion in all directions, whereas long and thin just gives you up and down mostly. So maybe that's actually a good strategy also just for approaching.
I know they're stimulating multiple of those ommatidia that are
Yeah,
detecting
yeah.
motion in the lot. Yeah.
And this one's interesting because whenever they do the leaf display, they don't come in straight. They actually come in doing this. So they actually have quite a lot of up-down movement. And I don't know whether they're trying to pretend to be a leaf that's floating, but it looks like it. Or maybe it's really trying to focus on this downward, upward movement rather than this expansion. I don't know. There's one in the back, sorry, Ines. Yeah. There's Rena.
I think the frequency are significantly better about the shape that it gets like that, like stay. I'm thinking about the background which you can see about and wondering whether the frequency of the water is similar to that frequency and whether they might be using a strategy of the background matching in terms of the one to react. But then I can't remember your results from the tracking experiment where you had that everything. Is then how similar in terms of the frequency and what the
Yes, but we were quite surprised actually, because we did spend quite a lot of time looking at frequency, and we were looking particularly for if they consistently change frequency as they're coming closer, maybe they speed up or maybe they slow down, maybe that might be part of it. We saw in that first example that I showed where the frequency was quite high, and when the prey escaped, the frequency dropped. So it's almost like they're kind of letting go of the pattern. But actually the frequency was really variable. Some animals really sped up as they were coming in, some slowed down, some were fast. This is actually one of the slower ones. Some were a lot faster than that. So the frequency is surprisingly variable, and we couldn't really find it correlated with anything in particular. We don't know whether maybe the frequency of the background might also be quite variable and they're matching that. That is quite a cool idea, but we don't really have a way of quantifying it below, beyond knowing what depth they're at, therefore how far away that is maybe. But yeah, the frequency is really interesting, really interesting. There's some suggestion that maybe the pattern might be driven by central pattern generators that are associated with fin movement. So we had thought that if the fins are batting faster, maybe then the stripes would go faster if they're really tightly linked. But it doesn't seem to be. Right? If they're in lots of current, they can, you know, the fins are going crazy, but they're still doing the same displays. So, but yeah, really good question. Yeah. Ines, I think you're next.
What's the success rate? I mean, do crabs ever escape?
That's a very good question. I mean, most of the time we're presenting the crabs in a way they can't really escape very easily. They do miss sometimes. They do misjudge the position of the crab. They also, they're very, very motion sensitive. So if the crab stops moving, they'll actually just completely stop. It's like they can't see it anymore. So they don't seem to focus in on the form of the shape of the crab. They seem to focus in on the motion of the legs. So if the crab stops moving, then they'll pause until they see something moving and then they'll start coming in again.
I mean, I just wonder whether the variation in tactic and variation in frequencies to stop the crabs learning.
Yeah.
Threat cue.
Yeah, so we just put in a little publication just reporting these 4 things. And one of the things we suggest is it's a frequency dependent thing. So if they all do the same strategy, then crabs will learn to avoid it, or they'll learn to associate it.
Only if they miss.
But only if they miss, exactly. Yeah. But the problem is we don't see natural feeding events very often. We do see them quite often attempting to approach an animal, which then swims away, and then they abort. So maybe that's an opportunity for learning. Yeah.
Just to follow up on that, sorry if I missed that, when you recorded these feeding events, did you know if it's the same cuttlefish coming back or not? We
did the best we could. We couldn't be 100% sure sometimes, but usually with the way that we sampled the reef is that we went along, it's a long linear reef, and we went along sampling animals, then going back and revisiting the same animals. They tend to hang out in the same place.
Could it be a personality thing and also an experience thing? Can you estimate their age or anything that, like, maybe they have to learn, given that this is a quite complex behavior and they're really smart, so maybe there is a learning curve for a cuttlefish, or maybe it's a personality thing. I just like to hunt this way.
So we know cuttlefish sex. You can tell the cuttlefish sex based on the coloration around the fins. We know the size of the cuttlefish, which isn't a perfect proxy for age because they do grow at very different rates depending on how much food they're eating. And we looked for correlations there with what displays they do, and we haven't found any. And you can revisit the same animal. Like I say, you can give it multiple feeding events and it'll do different displays every time. So they really do seem to either dial it up at random or dial it up based on some sort of local condition that we haven't managed to work out yet.
Back to the crabs, do the birds ever attack out of the sun or up
Yes.
degrees of the sun to minimize polarization?
Yeah, I think there's some quite interesting work on that. I don't know it very well, but there's I think it's the grackles, which are ground-based crab predators. It's a type of bird that they have in South Africa. I can't remember where it is. But anyway, it does this running movements through the colony, and all the crabs escape down their burrows. And they run, I think, from the direction of the sun, and then they do these curving movements to go and intercept crabs that haven't run down. So they're definitely using some strategies. The terns that hunt in our field site in Australia, they would always hunt into the wind because it means they could fly a bit slower and then they swoop down. So I think wind is also a factor. But crabs also have polarization vision, which they can potentially use to help reduce glare problems. I think we had some data that suggests that it's almost like inbuilt Polaroid sunglasses in their eyes that let them filter out some of that glare just to mitigate that. But yeah, attack direction is quite interesting. And we were quite interested in this as well, because there is a bit, especially when you're shallow, there's a bit of variation in the brightness of the direction of light that's coming. Maybe the cuttlefish were trying to attack from certain directions, but we didn't get anything concrete on that either. Oh, these cuttlefish were often depending on the current as well, so they would try and hunt into the current rather than have the current behind them, because I think it's much harder to control your approach when you've got a tailwind. Yeah.
Are they always successful? Like all the cases.
They're not, no, so sometimes they miss, and when they miss, it's usually quite dramatic, because we have the crabs on a little perspex base. If they miss, then they hit the perspex and the whole thing gets lifted off, and so you have like the camera and everything being taken off by the cuttlefish, and you have to chase it and try and get our GoPro back. So we've got quite a few funny videos of the
Not only for the camera, but
what, sorry?
any lessons learned, like.
By the
all
cuttlefish?
Yeah.
It generally took them a lot longer to come back again, put it that way. So I think they learned maybe to be a bit more careful. But I'd say 99% of the time, 98% of the time, they hit it first time, like they're superbly accurate. And I think part of the reason for this display is that it means that they can hover and really check out exactly where they're going to strike and really triangulate. So you've got both of these eyes kind of doing this binocular sort of depth perception thing. They don't want to be too close because they might injure their tentacles if they hit too hard. They don't want to be too far away because they'll miss it. So getting that distance just right is really critical for these animals. And so, yeah, the length of the tentacles also corresponds very closely to the eye separation as well. So the bigger the animal, the bigger the eye separation, the longer the tentacles. So I'm sure it's a nice geometric relationship there, the strike position. But yeah, it's really important for them that they really get that right, which is one of the reasons why they can't just bowl straight in and grab it. Yeah.
I mean, it's been great watching these videos over, and I love how you timed them all so they hit exactly the same time. I wanted to ask, you said that they get a bit smaller just before they hit, and I can see that watching the video. I'm not convinced it's that they're doing that on purpose. I think it's partly in order to get in, they have to release those fins a little bit to move inwards. It doesn't look like an act of getting smaller.
I feel like it's almost channeling also that strike tentacle. It's almost like they need to make that kind of cone for it to direct it, so it's maybe making it more accurate.
Yeah.
But we did think that maybe, you do get this gradual peeling in, and maybe there's something going on.
It looks like it's too late
for
the crab to do anything about it by that point.
So yeah, I think the missing link would be really nice to look at prey behavior in a natural context. That would be really great, but we have to work out a way of actually doing that in a reasonably controlled way. The nice thing is that these animals are great to work with in the wild, so you can just use them in their natural habitat. You don't have to bring them into a lab. It's all really naturalistic.
It's not such a bad place to go either.
Yeah, it's
Spend a lot of time underwater.
They learn if they do that, you're more likely to keep them.
Well, that's a good question. Yeah, there's been some suggestion that maybe at some very popular dive sites that they have learned to do that to divers, and they'll come and do the stripes on the divers. But this is very remote area, so I don't think there's a risk of that here. But we did find that if we went back to individuals too often, they would come to us when they saw us. So they're like little puppies. They learn very quickly that you've got food. So we tried.
That could work to your advantage.
It could, yeah, but we tried to sample as many different animals as possible to avoid that, and also so that we're not messing with their. We don't want them bothering tourists all the time by going and hanging out with them.
I mean, the fact that you said it was regionally very localized where they do this behavior immediately makes you think of some sort of cultural trait. But presumably these things never see an adult because they just live a year and die.
My suspicion is that if you house them completely independently from eggs, they would still do this. I don't think that they're learning from each other. They're not particularly social. They tend to. The other thing that I didn't show is that they use a lot of these moving patterns as social communication cues as well. So if there are two adults, two males near each other, they'll pass these pulses on when they're coming and approaching, and then they'll do these kind of zebra displays, a bit like the European cuttlefish. So maybe actually this is co-opted a bit from some of the social signals that they do also out on the barrier reef and everywhere else. They just haven't co-opted it in for their hunting.
You said that they're very motion sensitive, so could that be related that in order to be visible to the other cuttlefish, you want to be as moving?
I'm sure that's a part of it, especially in low contrast environments. You pass a dark, high contrast pattern across your head from a distance, they'll be much more visible than just hanging out. Yeah, I'm sure that's part of it. Yeah.
Any challenges in capturing the content with the cameras? And I remember in one of the socials we had recently, you want to build a motion capture kind of thing. But is it intended for both or just the cuttlefish or the prey as well?
In terms of trying to film the cuttlefish or
yeah, I mean, it could be really awesome if you could capture the whole 3D kind of cuttlefish motion, because then you could definitely model better
Yeah.
behavior, right?
So one of the things we did try to find a collaborator to help us with is to build a rig with multiple cameras to film it and then reconstruct in 3 dimensions. And it's like a virtual reality, actually play back in 3 dimensions, but then zoom the field of view right down to the crab eye so that you're really getting the crab eye view. We ended up abandoning that because we could get so close to it with the GoPro anyway. But it's only really at the very last minute that you find the perspective is slightly off. Actually, anything from back there, the perspective is probably close enough. I did think it would make quite a nice grant proposal, bringing together some virtual reality people interested in structure of motion and trying to reconstruct these things. I mean,
This presentations,
Yeah.
they are effectively to navigate.
yeah.
So I think that would be.
So Matteo's run I think is it called YOLO, the segmentation program. So he's run this and he can segment the cuttlefish completely from the background, the main video, using that approach. It works really well because it's often quite a simple background as well.
Yeah, no, that's really good idea, but with
Yeah.
implicit neural representations, you can actually navigate through the space.
Oh, yeah,
I mean, you could
yeah.
create 3D shapes
Yeah, yeah.
effectively with much less cameras.
Maybe we should
It's
have
a
that.
really-
That'd be really good.
Yes. I
That's
have to
using
work
AI
hard
to
in
sort of
that,
model
yeah.
the depth and then
Exactly,
shape
yeah.
perception. Yeah.
And could you go back to putting a mirror as well? And then you get right down behind the crab?
Yeah. I didn't thought of a
Yeah.
mirror. Go back to the old school PhD
Yeah.
kind of work. Yeah.
And then you get the 2 eyes of the crab right in the picture.
We actually brought with us a one-way mirror. So we spent a lot of time filming through one-way mirrors, trying to get them to react to their own reflection. They do some pretty cool stuff, actually. You can get them kind of signaling and so on. So yeah, it was really fun. Yeah. They have some quite exciting polarization patterns. One part of the story I haven't talked about is that they have these iridophores in the arms that generate polarized reflections. And what we think that's doing is it's potentially trying to reduce the polarization contrast of the background. So the background that they're doing this against has a polarization backdrop. And rather than just looking like a silhouette, like the birds did against the polarized sky, we think they might be reflecting a bit of light off these iridophores to try and approximate or just reduce that polarization contrast so that the actual looming kind of part of it is minimized in intensity and in polarization. But then the stripes are just this overwhelming downward motion. We think. So we're hoping we've got lots of polarization video of it, so we'll hopefully get some data on that soon.
Thank you. Should we continue over 1?
Thank you. Yeah, that'd be great.
I'm mesmerized. I feel like.